Good afternoon, everybody. It's really my great pleasure to introduce our speaker this Wednesday afternoon, Dr. Pamela Stanley. Pam currently holds the Horace W. Goldsmith Foundation Chair and is professor of cell biology at the Albert Einstein College of Medicine. Pam has had a remarkable career. Actually, when speaking to many of you about her pending visit, it was quite interesting to learn the sort of diversity of what Pam is known for, because each of you sort of came at it from a different angle. This isn't surprising because Pam's made many significant contributions to several fields of science. So some of you know her for her pioneering work using somatic cell genetics to isolate a highly instructive series of cell mutants that were defective in aspects of glycoprotein biosynthesis, the so-called CHO-LEC mutants, which could no longer bind to the lectins that they did when they were wild-type cells because of the altered sugar coat. Others of you know her work on spermatogenesis. And it's her team that demonstrated that conditional ablation of the gene that converts simple N glycans into complex ones results in male infertility. But most of you mentioned the work that I guess Pam is most well known for, and that is on Notch signaling. As many of you know, the extracellular domain of Notch receptors is decorated with carbohydrates, and Pamela's group is at the forefront of decoding which and how these sugars mediate Notch signaling. Pamela is a recipient of many, many awards, including a Merit Award from the National Cancer Institute and the Carl Meyer Award from the Society for Glycobiology. For her lecture today, entitled Glycans That Regulate Development and Notch Signaling, she will discuss Notch signaling, embryonic development, T and B cell development, and O-fucosylated glycans. Please join me in welcoming Dr. Pamela Stanley.
Okay, good afternoon, everybody. I am very, very pleased to be here to see all my old friends and meet new ones. It is a great honor to have been selected to give a WALS lecture. So thank you very much to Larry and Kelly for nominating me. And I hope by the end of my lecture that you will all feel a little sweeter, and that you'll understand that sugar is not just icing on the cake. So this is Albert Einstein College of Medicine in the beautiful Bronx. And glycans, which have been the focus of my work for a long time, became the focus because we realized that they form the frontier of every cell. So this is just a cartoon from Scientific American where you can see that anything that gets into the secretory pathway ends up on the cell surface, and that's where the glycans are mostly expressed. If we zero down and look more at what these glycans are, they're made up of different sugars that are depicted here. And so for the talk, I will be using these symbols, which will be defined on each slide. And they're added either directly to protein, as in this case, or they're added sugar to sugar. And so they can be fairly simple, just a single sugar here, or 4 sugars, or quite complicated, as in the N-linked glycans. There's more complicated ones, the proteoglycans, and so on. So to approach the functions of these modifications, we've taken a genetic approach. And the reason that we really decided to stick with it is that glycan expression is regulated. This is a tester section, and what it shows is glycans that are expressed in spermatids and spermatozoa that are not expressed in spermatogonia or spermatocytes. And since they're regulated, they must be controlled for some reason. And so our approach has been to use genetics. CHO cells, as Dr. Tabak mentioned, was where we began. And we have a number of CHO mutants, loss and gain of function. And I love anyone who wants to use a mutant to please ask me, because otherwise they just sit there. We've used them for glycosylation engineering, glycosyltransferase gene regulation, and to understand glycan functions, such as in lectin recognition, signal transduction, and the functions of dystroglycan in binding to laminin. And then in the 90s, we moved into the mouse and have made gene-targeted and transgenic mice, mostly glycosyltransferase mutants, again, to zero in on cell type specific functions of sugars. So let's just review what a CHO glycosylation mutant is like. If we take one, this is a complex N-glycan, and a loss of function mutant resistant to lectins could be LEC8, in which case all those sugars have disappeared and we have a new truncated form expressed on glycoproteins. Now, alternatively, we have a series of gain-of-function mutants, for example, LEC10, where we've acquired a new sugar that's expressed on glycoproteins. Now, the gene that codes for the enzyme that puts on this sugar is called MGAT3, and it's highly conserved in mammals, and it's highly regulated, and so we're very interested in what it does. And it first came onto our radar when we isolated a mutant that was resistant to ricin. And the reason for its resistance was the acquisition of this modification, the bisecting GlcNAc. And the enzyme that codes for, the gene that codes for the enzyme that puts this on was turned on in these mutants, and it's not on in CHO cells. Now, this has a pretty dramatic effect in that if we look at ricin resistance, LEC10 cells are about 10-fold more resistant to ricin. And however, they acquire at the same time an increased hypersensitivity to a different lectin, EPHA. And what these have in common is that they bind to GAL residues here. So what you can surmise is that the bisecting GlcNAc changes the conformation of these GALs so that now they are recognized very differently by these different lectins. And they also are recognized differently by galectins, which are animal lectins, and for LEC10 cells, there are a number of galectins which are decreased in binding to these cells. So what is the function of this very intriguing single sugar residue? One of the things that has been known about galectins is that they promote growth factor receptor signaling. And so this is just a simple diagram showing the N-glycans on receptors at the cell surface being cross-linked by a galectin, galectin 3 in this case. And so we were interested to see if something like the bisecting GlcNAc, which is not on these glycans, would affect growth factor signaling. And to do that, we set up wild-type CHO cells and then some mutants, including LEC10, and just expressed polyoma middle T and did a growth factor signaling assay using platelet-derived growth factor. And here we have representative glycans on the cells. So on a wild type, there'll be complicated complex N-glycans. In this mutant, we're missing one branch. In this mutant, we have no GALs. And in the LEC10 mutant, we have the bisecting GlcNAc. And what we found was that if there's really no GALs, we have greatly reduced PDGF receptor signaling. But if we have the bisecting GlcNAc, it's also significantly reduced. And same for losing a branch. So this is phospho-ERK1 over ERK1 and phospho-ERK2 over ERK2. So what happens in vivo? We knocked it out and used a mammary tumor model, the MMTV polyoma middle T model, to look at tumor burden and metastasis. And what we found is that if that enzyme is not there, you actually have an increased tumor burden, and the size of the tumors is also increased. And I'm not showing you the data, but metastasis to the lung is also increased. So it seems that the bisecting GlcNAc present in the control mice is basically retarding tumor growth, and like it did for growth factor signaling. So the bisected N glycans retard growth factor signaling by affecting their ability to bind galectins and their ability, therefore, to be actually endocytosed into the cells. So the galectin, the model here for the galectin lattice, is that these growth factor receptors are able to signal longer because they are not endocytosed as quickly as normal. And in fact, if we look into human data, relapse-free survival correlates with expression of this gene in this cohort that was published by Van Vert. So that is sort of an interesting side effect of having this bisecting GlcNAc is may be actually relevant to human disease. So there are a lot of CHO glycosylation mutants, and I'm just going to summarize them here. There's a number of loss of function mutants and then a number of gain of function mutants. And they've been useful to study human disorders of glycosylation by correcting the defects with wild-type genes and then showing that mutant genes cannot correct. In this case, LEC13, it's very useful because antibodies made in this cell line are about 1,000 times more toxic by ADCC. And as you know, many antibodies are in the clinic these days. In these lines, which make truncated glycans, enzyme replacement therapy has been helped because they target glycoproteins with those sugars to the reticuloendothelial cells and greatly facilitate enzyme replacement therapy. These sorts of mutants make selectin ligands, and so they can target glycoproteins to cells that are expressing selectins. And as I mentioned, LEC10 is important for growth control. And then mutants that are lacking sialic acid have been very helpful in understanding the receptors of pathogens. So we have also used these mutants to aid in the discovery of new orphan-like glycosylation activities. So genes that are discovered in the genome that look like they might be a transferase or a glycosidase. And this is just to show you that if you cleave off all of the N-glycans from CHO cell glycoproteins, you find that there is a bunch of oligomannose N-glycans and then many, many different complex N-glycans. And for example, down here, the highest molecular weight one has a structure like this. Now, since we're only talking about mass, we can't actually say what the structure is, but it is quite large. And we decided to use this approach to study an orphan cDNA which had been isolated at Riken. And when we expressed that in wild-type CHO cells, this is the pattern we got. There were absolutely no complex N-glycans. And we overexpressed it in wild-type CHO cells to show also that the cells became very resistant to the lectin LPHA. So they're not making complex N glycans, which is why they're resistant to LPHA. And they've got a increased amount of this species, which is MAN5. So MAN5 is the substrate for this enzyme MGAT1, or also known as GlcNAc-T1. And this cDNA encodes for a protein that's a typical type 2 membrane-bound type protein, and it inhibits MGAT1. So you can see from this, MGAT1 catalyzes the transfer of GlcNAc to this sugar, to this substrate, and if that reaction doesn't happen, the whole pathway is inhibited. And so this substrate accumulates, and there are no complex N glycans. Now, this was very interesting because it's the first sort of physiological inhibitor of a glycosyltransferase like this. We've previously, other groups have found chaperone or other activities, but not an inhibitor. So we showed that it inhibits the enzyme activity if you make an extract of the transfected cells and co-IPs with the enzyme. And so we've been looking more directly at specificity using a FRET assay in collaboration with Sakari Kelakumpu in Finland. And the idea here was to see if MGAT1 was the only GlcNAc-transferase in the medial Golgi that would interact with this inhibitor. And so the inhibitor, which we call GlcNAc-T1 inhibitory protein, and it's L for the long form, has a monomeric venous GFP attached at the C-terminus and is stably expressed in these lines. And each one of these transferases was conjugated with M-cherry and then transfected into the line. And when we did that experiment, FRET interactions were seen either in COS7 or LEC1 CHO cells only with MGAT1. Other GlcNAc-transferases in the medial Golgi did not cause a significant FRET signal. Now, this was true if they were co-transfected together. And we can see that the inhibitor actually forms homomeric complexes as well, as do each of these enzymes. And you can inhibit this interaction if a complex is formed by overexpressing individual MGAT1, cDNA, or the inhibitor, but not any of the other GlcNAc-transferases. So it seems to be very specific for MGAT1 in the medial Golgi. We've also shown that if you put it into the endoplasmic reticulum, it doesn't inhibit MGAT1 and it doesn't inhibit other Golgi glycosyltransferases. So what is it doing? Well, we were pretty surprised when we had a look at where it's expressed and found that it's very highly expressed in testis. It's expressed minimally in other cell types. And so we looked at the expression specifically of the L form and found that that was developmentally regulated. At 22 days, the transcripts for this form were higher than 11 or 17 and higher than the adult. So this was intriguing, and we have just seen some recent data, analyzed some recent data from RNA-seq, where the MGAT1 gene is shown here in red in Sertoli, Spermatogonia, spermatocytes, spermatids, or spermatozoa, and you can see that it's expressed in all of them at differing levels. But the inhibitor is expressed in spermatocytes and spermatids only. Now, I've put here that it's called MGAT4D because the Human Genome Nomenclature Committee has now named it MGAT4D. However, there's no evidence that it's a transferase at this point. Now, it could be. It could be an inhibitor and a transferase or whatever, but that's its new name. However, I'm going to keep calling it GNT1IP. So, we were also interested in analysis of human testis biopsies where the transcripts for GNT1IP and MGAT1 were determined. And these are men with tubular atrophy or Sertoli cell only syndrome, spermatogonia only germ cells, meiotic arrest of primary spermatocytes, so they would have spermatogonia and spermatocytes, and then decreased spermatids or just everything being normal. So in the normal situation, MGAT1 is quite poorly expressed and the inhibitor is highly expressed, and so on all along. So over here in tubular atrophy where there's no germ cells, the inhibitor is not present and the enzyme transcripts are well represented. And so they're completely complementary. So that was sort of an interesting aside. And our model at this point is, or the summary of this data are that if we look at the transcripts, we look at spermatogonia, spermatocytes, spermatids and sperm, which are all generated on the Sertoli cell during differentiation until the sperm go into the lumen, that the enzyme is expressed highly in spermatogonia. So we expect complex N glycans here. Then transcripts for this go down a bit, and these ones come on. So we expect much more representation of this N-glycan. And same here, although probably a bit more mixed. And then in sperm, we have the inhibitor not represented and the enzyme there. So are these working in a complementary fashion, and are they needed in a complementary fashion during spermatogenesis? Could these high mannose N-glycans enhance Sertoli germ cell adhesion? So we haven't done too much on this, but we have done one series of experiments where we used TM4 Sertoli cells, which are here shown attached to a dish, and they're actually fixed. And we took cells expressing the inhibitor, two different forms of it, and found that if you allowed them to attach and then washed 5 times, that these cells remained quite strongly attached to the Sertoli cells, whereas the control cells expressing complex N glycans did not. And a mutant that's missing the enzyme altogether had the same result as the cells expressing the inhibitor of the enzyme. And these data are just shown quantitated here. So it could be that the inhibitor is designed to really decrease MGAT1 activity after the transcripts have been reduced. Any enzyme that's remaining will be also reduced by the inhibitor, and that may enhance the interaction of these germ cells with Sertoli cells. So that is our working model. And what happens in vivo? We have deleted the enzyme MGAT1 in spermatogonia using a CRE that is expressed only in spermatogonia at 3 days of gestation, 3 days after birth. So what is shown here is the whole testis, and then a testis tubule, and then a blow-up of differentiation from spermatogonia through to sperm on the Sertoli cell. So if we delete MGAT1 in these cells, what do we find? And we can show that if we look at the expression of complex N glycans with the lectin LPHA, they're expressed nicely here in which is expressing the Cre in spermatogonia. And what we found is that for wild type, this is just testis sections stained with H&E, you can see all of the germ cells as they're differentiating into the lumen of the testis where the sperm are congregated. And we found that in these mutant mice where MGAT1 is knocked out in spermatogonia, that things look pretty reasonable up to spermatids. And then the spermatids fuse and form these multinuclear cells, which are just syncytia, really. And of course, this blocks spermatogenesis, and we can see that clearly if we look at the epididymis, which actually contains no sperm compared to the control mouse. And this is just a much older mouse, which now has all sorts of things collecting in the epididymis, but no sperm. And so we are trying to work out the mechanism behind this. We're looking at microarray data and proteomics of germ cells at 22 days before they show any of these phenotypes to see what pathways may be mainly affected. We are knocking out the inhibitor in the mouse, and we've also generated a number of transgenic lines that overexpress MGAT1 or the inhibitor in specific germ cells using promoters that are active only in spermatogonia or spermatocytes or spermatids. So we hope to be able to show that this inhibition of MGAT1 has really a functional role in spermatogenesis. And now I'd like to turn to the third part of my talk, which is on Notch signaling and roles for glycans in Notch signaling. So this is a familiar diagram, I'm sure. Notch receptors are transmembrane. They're heterodimers linked together non-covalently outside the cell. And the N-terminal domain has many EGF repeats. In the case of Notch1, it's 36 EGF repeats. And Notch signaling is really a cell-cell signaling event where an adjacent cell expressing a ligand, either delta or jagged, interacts with Notch. The ligand interacts with Notch through the DSL and N-terminal domain. And that causes a conformational change that allows this metalloprotease to cleave the extracellular domain, and it gets endocytosed into this cell. Then the gamma secretase can release the intracellular domain, which goes into the nucleus and in complex with co-repressors ends up activating many downstream target genes. So the main thing is that Notch EGF repeats regulate this event, and they are coated with sugars. So this is mouse Notch1 extracellular domain with 36 EGF repeats, and each EGF repeat is about 40 amino acids and has 3 sets of disulfide bonds that are linked through these residues. And the sugars are added at a consensus site. So the O-glucose glycans are added between cysteine 1 and 2. The O-fucose glycans are added between 2 and 3. And the O-GlcNAc are added between 5 and 6. So each of these is an EGF repeat, and then we have glucose, xylose, xylose. So they're simple, but there's many of them, and there's 3 types. So some EGF repeats can theoretically have the 3 types of these modifications. And we have focused on the O-fucose glycans, and originally wondering, do they regulate Notch signaling, and then how do they regulate Notch signaling? And the first experiment we did after realizing that fucose was actually the first sugar in this glycan was to delete the enzyme PO51, and found that if there's no possibility of adding an O-fucose glycan to Notch, that it's embryonic lethal, that there's a severe vascularization defect of the embryo. This is the yolk sac, and here is the blood all pooled. That there are, the somites are not properly formed. Here in the wild type, they're very organized, and here they're very lumpy. That Notch target gene expression is altered. So here, delta 3 expression is reduced in the pre-Semitic mesoderm, but enhanced in the brain. And we looked at many different Notch target genes, and the embryos are severely retarded in their development. So what's the mechanism? The phenotype copies global loss of Notch signaling, but is it due to an effect on Notch ligand binding? And we looked at that in ligand-dependent Notch signaling assays, where we used CHO cells that have endogenous Notch receptors and co-cultured them either with control cells or with those cells expressing the Notch ligands. And then transfected with reporters, firefly luciferase here, which is driven by the upstream RBPJ kappa binding sites, which will only respond to the intracellular domain of Notch receptors. And so this is a co-culture assay, the readout being fold induction of luciferase in ligand-expressing cells versus L cells. And we had a mutant that was able to help us understand if fucose itself was important or if the enzyme was more important. And this mutant is called LEC13, and it's defective in the first enzyme of the endogenous pathway to make GDP fucose. So in the cell, this is how GDP fucose is made. And this just shows that in a LEC13 cell, this lectin cannot bind. Now, you can also correct this defect by adding fucose externally to the cell. It will be taken up, converted to fucose 1-phosphate and GDP fucose. And so we have a mutant and a rescue situation. And we can see here that in a control cell stimulated with L cells, there is not much reporter activity. Stimulated with delta 1, we have good fold stimulation, and that this is inhibited by a Notch inhibitor, a gamma secretase inhibitor, if that's included in the medium. So we know it's dependent on Notch. And in LEC13, we have very little stimulation. So the fucose itself is actually doing something here, because as I can show here, this is just the same sort of thing where we've rescued with fucose or galactose as a different control sugar. And you see that fucose can rescue LEC13 for delta 1 stimulation and for jagged 1, and gal cannot, and that the control cells don't care less about fucose. But most importantly, the level of the enzyme is the same in all these cell lines. So there is a little bit of discussion about whether it's a lack of fucose or the lack of the enzyme. And in mammals, fucose is important. In Drosophila, it seems that the enzyme itself may also have some functions in getting Notch to the cell surface. But in our case, we could look at binding of O-fucose glycans, of the ligands to Notch receptors on ES cells to ask that question. And we found that the mutant ES cells lacking the enzyme have very little binding of delta 1. This is soluble delta 1 compared to the control ES cells. And this is dependent on calcium, which is a feature of Notch ligand binding. It must have calcium to exhibit good binding. And it is true for delta and jagged Notch ligands. And most importantly, if you look at the expression of Notch receptors at the surface, they are there in equivalent amounts in the wild-type ES cells versus the mutant ES cells. This is Notch 3. We've looked at Notch 1 and Notch 2 as well. So the fucose itself is important. What about an individual single O-fucose glycan? So here we've got the diagram again showing where O-fucose glycans would be, and we made a mutation just in EGF12, which is in part of the ligand binding domain of Notch receptors. So this is the only O-fucose here, and the surrounding EGF repeats don't have the consensus site for O-fucose. And we could show that this mutation, threonine to alanine, eliminated fucose incorporation using this assay. And so what happens in the mouse? Well, the mouse was fine, actually, but it actually grew slightly more slowly than wild type. But we decided to look at T cell development because Notch is known to be very important in T cell development, and found that the thymus was smaller in the homozygous mutants, the thymus weight was smaller, and the absolute number of thymocytes was smaller. And when we looked at the different T cell subsets during differentiation from early thymic progenitors through the double negative 1 to 4, double positive and single positive T cells, so named because of expression of CD4 or CD8, then we found that a number of the T cell subsets were really affected. Double positive T cells were reduced about 50%, intermediate single positives also, and the single positives were also reduced. But other cell types in the thymus were not altered. And this phenotype was cell autonomous. In other words, it had to be reduced in the T cells of the mutant, and if we put that into a lethally radiated recipient, the control cells would produce thymocytes in reasonable numbers, and the mutant would still only produce half as many. And that's thymocytes, and the same for DP T cells. So a single point mutation in the Notch receptor at the O-fucose site affects T cell development. And it's important to have this modified for optimal T cell development. So there's another sugar that's added after the fucose, and the next one is GlcNAc, N-acetylglucosamine, and it's transferred by enzyme that was called Fringe because it was discovered in Drosophila, and when it was overexpressed or actually over ectopically expressed, it formed a little fringe on the Drosophila wings. So this fringe gene, or one of the fringe genes, had been mutated in the mouse before it was even known what it did. And two groups showed that you get tremendously disrupted skeletal development in these mice. So we were able to show, and other groups too, that Fringe modulates Notch ligand binding. So here we have cells that are binding soluble delta-1 ligand. If you overexpress lunatic Fringe, they bind better. Whereas if in the same cells, binding of jagged-1 is decreased. So Fringe is a modulator of Notch ligand binding, and an important one. And in this case, with lunatic Fringe, it's really needed for somatogenesis. Now, lunatic Fringe was also shown to affect T cell development, and it's expressed during the early phase of T cell development. It's turned off in double positive cells, and it comes back on again in the single positive cells. So two groups showed that if lunatic fringe is knocked down, fewer DPT cells are made, and they compete poorly with wild-type cells in transfer experiments. And also, if lunatic fringe is upregulated in these cells, so misexpressed in these cells, they compete with the immature cells and end up forming B cells in the thymus. So fringe was known to be important, and the question was, what about the other fringes? So there are actually 3 fringes in mammalian cells. They're not all expressed in the same cells, and if you knock out manic fringe or radical fringe, it doesn't affect skeletal development. However, in both T cells and B cells, transcripts are present for all 3 fringes, albeit at different levels. And so we decided to see if we could tease out functions for each of these fringes in the different T and B cell development situations. And the question was, are they all functional? Are they redundant? Are they complementary? So to do that, we wanted to generate fringe triple knockout mice. And we were able to obtain from Susan Cole fringe triple het mice, which have only 1 copy of each of the fringes. And so when you cross them together with this one, which is double mutant for manic and radical, but expressing lunatic, you could generate mice that are missing 1 of the fringes or 2 of the fringes or none. And from those mice, the fringes that remain are either 2 of them or 1 single fringe or no fringe. So we decided to focus on this group to see if there was a phenotype when all the fringes were gone, and if we could rescue with these single fringe expressing mice. Now, actually, I'd mentioned that lunatic fringe has a phenotype. It doesn't have a phenotype in adult mice. It was only if they looked at very immature fetal cells that a phenotype could be observed. So the mouse compensates if you just knock out lunatic fringe by itself. And nothing much was known about radical and manic. And so we compared ligand binding for double negative T cells, the immature T cells, in the triple knockout, and found that binding of delta 4, which is the ligand expressed on the stroma, is significantly reduced. And however, binding of Notch 1 and Notch 2, or antibodies against Notch 1 and Notch 2, show that they are not reduced. So this is not because Notch is not getting to the cell surface, but to do with presumably the sugars on these Notch receptors. Notch 1 is the predominant Notch in T cell development. Notch signaling was reduced in the double negative T cells lacking fringe based on these target genes being reduced. This is deltax1, both of these, and CD25. So this is a fairly minor effect, but it was significant, and this is a known Notch target gene in these T cells at that stage of development. And when we look at T cell subsets in the absence of fringe, we found not anything dramatic with double negative cells, but double positive cells are reduced, and single positive cells, interestingly, were increased. This is the opposite of what we saw when we knocked out the O-fucose. And no significant differences in the other cell types in the thymus. Now, in the spleen, it's been shown that lunatic and manic fringe cooperate with each other in facilitating the differentiation of newly formed B cells into marginal zone precursors and then marginal zone B cells, into follicular precursors and follicular B cells. And whereas in thymus it's Notch 1 and delta 4, here it's Notch 2 and delta 1. And so, however, nobody has looked at radical fringe in this situation. So in the triple knockout mice, the weight of the spleen is reduced, and the numbers, or the frequency, not the numbers, but the actual proportion of CD4 and CD8 T cells are reduced. Now that's quite interesting because in the thymus they were actually increased. And so we're now looking at these to see if their activation state is altered. In the generation of marginal zone B cells and follicular B cells, one can identify those using these different markers, CD21 and IgM. And follicular B cells were slightly increased in the triple knockout, whereas the marginal zone B cells were significantly decreased. And so now there we are with the triple knockout having effects on both T and B cell development, but no effects on the other cell types, myeloid or B cells. And now we looked at the individual singly expressing fringe mice. So this is the triple het control. This mouse is expressing only radical fringe, or only manic, or only lunatic. And we can see that in the cases where we had an effect in the triple knockout, it has been rescued by each of these fringe enzymes, both for the double positive T cells and for the single positive T cells. In the case of the spleen, it was largely rescued, but in some cases not completely rescued by the single fringes for the T cell numbers. Follicular B cell numbers were normal, and the marginal zone B cells were rescued by lunatic and manic, but not too much by radical. And so radical does not seem to interact very much with Notch pathway in the marginal zone B cells. So there are a number of key questions to address from these data, and one of them is, does Notch 1 carry only O-fucose in double negative T cells lacking fringe? So if this is all not there, is there a substitute there? Because as you saw, the delta 4 binding was very low. However, we do get a lot of T cells. So potentially there's another sugar added or another enzyme that can add GlcNAc, and that is something we really want to find out. We want to find it out in vivo because Notch pathway regulation is very sensitive to changes in expression level. So we have thymuses from these mice, and we'll be doing a mass spec analysis, hopefully, on Notch 1 in collaboration with Bob Halterwanger. Can a different sugar substitute for fringe? That would be another question. For example, in Drosophila, there's a glucuronic acid attached to fucose, and that's never been seen in mammals. Is there another GlcNAc-transferase? Are the Notch 1 O-fucose and other glycan sites, so all these putative sites, are they actually occupied, and does loss of one sugar type affect the expression of one of the other sugars? We don't know. And then which Notch 1 sites are modified in double negative T cells that are expressing only lunatic, only manic, or only radical? Can they really substitute totally for each other, or is there a subset of sites that are needed for T cell development? And finally, we hope to tease out roles for each of them using the single fringe mice in co-culture assays of T cell development, where you can use each ligand separately and look at how far cells progress during development. So to summarize then, the O-fucose glycans and Notch signaling, what we know is that if we miss one O-fucose glycan here, we affect T cell development. Glycosyltransferase knockouts have not been studied for Notch signaling, so we don't know, but there are 6 potential ones that could modify it. GALTs have been looked at. My lab looked at GALT1, and there are mild Notch defects, but again, there are 6 beta-4 GALTs, so they could be much more important than that. And then the knockout of all fringes would give you theoretically this, and we know that lunatic gives skeletal defects and that each of them contributes to T and B cell development. And if you knock out PO51, you end up with none of these sugars. So this is a global Notch signaling block. So which sites are actually modified and how in vivo are key questions. And what are all the essential players that are required to generate these? Because each of these enzymes needs to get its substrate, GDP fucose, there are transporters and so on, and other activities that contribute to the final product, which we are also trying to identify. Now the last glycan, the most recent glycan on Notch, was discovered by Tetsuya Okajima and is an O-GlcNAc. And you've probably all heard of O-GlcNAc, especially here, but the O-GlcNAc that is worked on mostly at NIH is an O-GlcNAc in the cytoplasm, and this is O-GlcNAc added in the secretory pathway and to an EGF repeat that has to be folded properly. If you denature the EGF repeats, none of these enzymes work well at all. So we decided to look at this in the fly because there are many transcripts in the mouse, and looked at knockdown in the Drosophila wing using an engrailed Gal4 promoter. And if we knock down the enzyme that puts on this O-GlcNAc-transferase called EGF O-GlcNAc-transferase, we get wing blisters. And we showed that if you genetically examine interactions between this knockdown and Notch pathway mutations, that they do interact and Notch mutations suppress the blisters. And interestingly, just last 2 years ago, a human which had completely lost EOGT activity was reported, and the syndrome was termed Adams-Oliver syndrome because it's very similar to other genes that give rise to Adams-Oliver syndrome, the phenotype in this person. So altered digits and calcium deposits in the brain in this particular case. But there are effects in the skin and so on. So they're quite suggestive of Notch signaling, and we are now investigating a mouse that hopefully will allow us to identify exactly what this EOGT enzyme does. So, in fact, congenital diseases from reduced O-glycans on Notch have been published quite a bit in the last year or so, as well as before. So for O-fucose, there are autosomal dominant mutations in PO51 that were published in 2013, and that gives rise to Dowling-Dagos disease. Again, a known syndrome, but this is a new cause of it. I mentioned the mouse situation with skeletal problems, but there's also a human lunatic fringe mutation giving rise to spondylocostal dystosis. And then for the O-glucose glycans, this enzyme actually can add glucose and xylose, either or. it gives rise to the same sort of syndrome as Dowling-Degoss disease. It was published last year. And then the Adams-Oliver syndrome for loss of this particular O-GlcNAc. So it's pretty clear that Notch needs sugars on its extracellular domain for various things, including ligand binding, and in other cases for the stability of Notch at the cell surface. And those of us who are interested in this are really trying to nail down mechanisms by which this all works and be able to define at a molecular level what's happening. There is a very nice crystal structure by Chris Garcia of delta 4 and Notch 1 domains showing that the O-fucose is right in there and needs and coordinates with amino acids. And actually he concludes that it's like the 21st amino acid in that situation because it's allowing the interaction with delta 4 to be much better than if it's not there. So now I'd just like to close by saying that it all began with a simple question, and my question was, is lectin resistance stable and heritable? Nothing to do with glycans. And it started when I picked a colony and put it in a tube and then characterized everything that came out of that colony, which were many, many, many things that were certainly not on anybody's mind, and especially mine, when I picked the colony. So I think just basic science, unforeseen rewards is how I would like to conclude. And I'd like to thank, of course, all the people who have contributed to this work. These are recent people from the lab who contributed to most of the work that I mentioned today. And my funding from NIH, both NCI and NIGMS, for many, many years. And so I thank them, because without that I wouldn't have done any of this, from Einstein and various foundations. And then there have been many collaborators over the years who are listed here, some at Einstein and many elsewhere, and others in the lab, each of them known for the mutants that they characterized. And now there are people carrying on with Notch and Fringe's MGAT1 and the MGAT1 inhibitor at present. So I thank them all, and I thank you for your attention.
Thank you. So we have time for a few questions. So this is a special time at NIH. We rarely serve cookies, but for this lectureship, afterwards there's cookies and beverages, I guess, out there through that door. They're the library, and so this is the only thing standing between the cookies. John, why don't you start?
Sorry, the carbohydrate people, you know, we're unstoppable there. So Pamela, very nice. I have a quick question about the spacing. You know, the evolution of the EGF repeats as gene duplication events. Is there anything unique about the spacing of the individual glycan sites, and is it how conserved? Do we know how conserved that spacing is?
Some of the sites are very conserved, like EGF12 is highly conserved, and some of them are not so conserved. So off the top of my head, I don't know. I mean, probably about 6 or so O-fucose sites are highly conserved. But EGF12 is definitely very conserved. Very conserved. Yeah. And the spacing, I mean, all of those diagrams are totally theoretical because they are the predicted, I mean, they're based on the fact that the consensus sequence is there. There could be sites which don't use exactly that consensus, or some of those may not be used ever. That's why we really want to do it in vivo, from thymus, Notch1, in mice, that have never been perturbed except for having a mutation.
And if no one else has a question, a related question about the
There's
stem
one
cell
there.
niche, about the stem cell, do you think, based on this spermatogenesis result, there'll be unique inhibitors for each of the stem cell niches?
Oh, I don't know. I mean, I think that I doubt it, but this thing, we were looking at it because it was like a transferase and it turned out to inhibit. So this is an artificial situation. We've overexpressed it in cells. But then you look at where it's expressed, and it's a very specific sort of site of expression in the testes. So whatever it is, we'd like to get to the bottom of it. But whatever cell we put it in, either co-transfecting with MGAT1 so they're equally expressed, or putting it in to inhibit endogenous, in different cell types it inhibits MGAT1.
I was just wondering, for such a complex system and such a complex code, what is known about the synthesis and the addition of these different sugar moieties, and specifically with the editing of it, because it appears that a simple mistake in even one of those repeats could have very detrimental effects.
Yeah, well, the synthesis, the transferases are all known, pretty much.
I meant for Notch and how they're added, sorry.
Sorry, what?
I meant to say how these are added to the Notch, EGF-like repeats, yeah.
From glycosyltransferase. So the O-fucose is added by PO-fucti1 and GDP-fucose. So both have to be there. That's the substrate. So there's an enzyme dead, PO-fucti1, and in the fly, that could largely rescue aspects of it, although there's a more recent paper that says there are still problems. In our situation, in mammals, we could sort of rescue a little bit, not very much, with an enzyme dead PO-fucti1. And if we did an irrelevant enzyme and overexpressed it, we got the same amount of rescue. It was as though overexpressing in the ER was not good. So if you look in mice where PO-fucti1 is knocked out in bone marrow, in hematopoietic stem cells, or in myeloid cells, it's work of Lan Zhu and John Lau, there's a little bit of decrease of Notch signaling, as though the enzyme itself does help Notch get to the surface. But so for each of those enzymes, so that last slide with the human diseases, that's what's known. Is there something else that you were asking? No?
One final question.
There are these three homologs of Fringe. Do they, O-gluconate-focus, same sites or different sites? Is there anything known about that?
Well, that's the question. And that's what we want to address with the single fringe mice and the triple knockout mice. But if you look at them, like Bob Holterwang has done experiments which have been reported at meetings where overexpressing each fringe by itself will give some variation. So they may not equally do every single site.
The selection is probably selective for some sites, each one of them.
But again, this is often with pieces of Notch and so on, and overexpression. So it's not that, yes, overexpression of the enzymes. So I think it's important to do it under physiological conditions.